Good afternoon, everyone. Welcome to Research and Discovery Grand Rounds. We're just going to give a few minutes for everyone to join the call, so we'll likely wait until a couple minutes after to get started. Okay, it's 2 minutes after, so I think we can go ahead and get started. Welcome, everyone, to Research and Discovery Grand Rounds. My name is Bridget Jones. I'm a professor of pediatrics at Children's Mercy, and I co-lead translational endeavors within Frontiers. So I'm very happy today to introduce our speaker. So we're joined today by Dr. Diego Mazzotti. Dr. Mazzotti is an associate professor and director of the Division of Medical Informatics in the Department of Internal Medicine at the University of Missouri-Kansas Medical Center. His work sits at the intersection of biomedical informatics and clinical research, where he applies advanced data science and machine learning methods to large-scale physiologic, genetic, and clinical data. Dr. Mazzotti has trained internationally, earning his PhD in psychobiology from the Federal University of Sao Paulo and further specializing in biomedical informatics at the University of Pennsylvania, where he also held research and faculty positions. His research focuses on translating complex, high-dimensional data into actionable insights that improve understanding of human health and advance precision medicine. And so today he will share his insights from his work through the NIH All of Us research program data set and discuss the role of big data in shaping the future of precision medicine. So please join me in welcoming Dr. Mazzotti. So thank you, and I will turn it over to you.
Yeah, thank you so much, Dr. Jones. It's a great pleasure to be here today in the last session of this Frontier Grand Rounds, the spring session, which, for those of you that join all the sessions, I think we learned a lot about many of the resources that are available in this area across all of our institutions. And I'm glad to kind of close this with talking a little bit about the All of Us research program, which one of the projects that I'm very excited about. And we have lots of folks, students, faculty, staff working on many of those many projects on using this data set. So here's a brief outline of what I'm planning to speak to you today. So I'm going to talk about the All of Us research program, what it is, how it works, what data is available, and I think that hopefully we will instigate some ideas for your own interests in providing and proposing a study using the data set. The All of Us is not only about data. I think there's a lot of participant trust, and I think I'll mention some of the aspects that the All of Us research program is doing to engage with participants and then create and enable this trust. And also, if you were interested, if you haven't joined All of Us and access the data, I'll just show you the simple steps that you have to do. It's freely available. If you're part of one of the institutions that have a data use agreement with All of Us, you are automatically available to access this data. And then I'm going to move on to some of the studies that we are conducted here at the University of Kansas Medical Center that pretty much our team, not only my group, but other collaborators and investigators at the University of Kansas Medical Center working on sleep, Alzheimer's disease, wearable social determinants of health, and cancer. And also some opportunities to get involved if you want to learn more, right? So we're going to talk a little bit about a training program that I actually had just completed. We're in our last week, next week, but we are planning on releasing this program on an yearly basis. And also how to join all of us as a research participant. So, all of us is for all of us. I'm all of us research participants, so I'm hoping to also provide some insights of how you can participate not only as a researcher, but also as a participant. So, what is the NIH All of Us Research Program? So, the research program is this historic longitudinal effort to try to collect data from at least 1 million individuals living in the United States, right? So, the goal is to come up with a biobank that links different sources of data with the purpose of accelerating health and research and improving health. Right. So instead of being focused on a single domain, the goal of all of us is to be comprehensive. So it tries to understand individual differences in lifestyle, in socioeconomic contributors, environment, and biology. So then we can go towards the process of understanding precision medicine and understanding healthcare that's solely for each individual. So the All of Us Research Program is part of this broader precision medicine initiative. And I think they really have this kind of idea of if we are all biologically unique, why medicine is not one size fits all. So that's not really a true statement, right? We are variable. We have biological variability. We have physiological variability. We live in different environments. So the All of Us Research Program is trying to collect data across this variability to try to understand how we can get the right treatment to the right person at the right time, right? And up until the All of Us program, there were not any very large and diverse longitudinal data sets. We have smaller data sets, maybe from electronic health records or from some other biobanks in other parts of the world, like the UK biobank is very popular. But many of those biobanks were also restricted to more homogeneous populations. And I think what all of us brings us is this ability to provide diversity across the board, not only biological diversity, social diversity, and pretty much regional diversity as well, geographic diversity. And so that's really the solution that all of us is trying to bring. So the mission of all of us, as I said, is to accelerate health, enabling individualized prevention and treatment and care for all of us. And they really participate into those three steps, right? So they have to nurture partnerships for decades because 1 million participants is no small feat, right? So we are trying to not we, but all of us research program are trying to engage with participants from the very beginning as prior to recruitment as they enroll participants to the program and how you engage with participants throughout the process because that's if participants are trusting the process of participating in a research study at that scale then you can obtain more trustworthy data and that can reflect really the reality of the United States. One of the goals is to then deliver this largest and richest biomedical data sets that's available. And we're going to talk in more details of what are the data elements that are there for this program. And it is, in fact, the largest and the richest biomedical data set that's available. And also, not only answer scientific questions of the data, but there's so many more components to being within this ecosystem of all of us because there are researchers that are developing methods and strategies to analyze this data in ways that was not possible before because all the other studies were at a smaller scale. So the all of us is creating ways of how researchers can share data or can share their code to analyze the data in a format that makes it easier to make scientific dissemination faster, scientific reproducibility more reliable. And I think we are all, as a community, learning and improving through all the processes that all of us is giving us. To give you a little bit of background on the timeline of the program, right? So the program has been happening now for over 10 years. And I think that the initiative, which is started as the Precision Medicine Initiative, really was announced in 2015. And that's when after, that initial announcement, a pilot study occurred where about 5,000 participants were enrolled. And then over the years, several more participants were starting to enroll all over the place within the United States, right? In about 2019 and 2020, that's when the Researcher Workbench launched. So the Researcher Workbench is this platform that researchers can access the data and perform the analysis and publish their manuscripts. So after this initial data release, that's when researchers started to ask, address questions that they have with the data set that was available. But perhaps the greatest data element that really makes this data set very unique is the genomic data. So the genomic data became available now for several participants towards the earlier 2000s, and it really has impacted how we understand the relationship between genetic factors and all sorts of diseases in the context of known genetic variability across populations, which is really something that was not, available in the literature, as most of genome-wide association studies were focusing on populations of generally European ancestry. So with the All of Us research program, now we have the genetic variability to start looking at specific genetic ancestry groups, or even admixture, and something that was not quite possible at that scale. And more recently, there is an initial launch of pediatric enrollment, which I think it's probably one of the most exciting news as far as those large biobanks, because I don't believe that there are any other biobanks that are incorporating pediatric data. So the All of Us research program is a large national program, but here at the University of Kansas Medical Center and some of our neighboring institutions, we are lucky to be part of what is called the All of Us Heartland Consortium, which is a partnership between academic medical centers, hospitals, and community organizations in the central U.S., in enrolling areas from Kansas, Missouri, Nebraska, and Iowa, which is really try to incorporate and increase the recruitment for participants in the All of Us in this area of the country. So our role, the KU's role in the program is to serve as the lead institution of the Heartland Consortium, and the PI is Dr. Ojo, or Dean Ojo. And that's kind of like our timeline for enrollment. So the funding was announced in 2023, and in November 2025, which is about 6 months ago, that's when we had the initial family and pediatric enrollment began. And then now we have Children's Mercy as one of the partners within the All of Us Heartland Consortium, which we're hoping to really ramp up the recruitment from pediatric population. So this is the research team for the Heartland Consortium, and it's very exciting to see all of those investigators working together to ensure that we have, we have our role as our institution and partner institutions within the All of Us. So just to, again, even prior to enrolling participants that are pediatric, which I think that there is a great, process of, informed consent and assent. We have to enroll not only the pediatric patients, but the family. But because of the EHR data that is available for those participants that decided to share the electronic health record data with all of us, is their historical data. So participants that were part of health systems that have agreements with the All of Us and ended up being pediatric participants prior to their enrollment, the data is currently available. So there are lots of studies going on across the board, not only at KU, but across the board on pediatric data. And then here are some of the publications. So if you're interested in learning more about the use of pediatric data in all of us, those are great initial literature that you can start looking at until more participants are actively recruited for pediatrics. But I'm not a pediatric researcher, so I'm not going to focus on most of what I'm going to be talking about here today on that. But I'm excited to show a little bit more about the population and the data that is available within this program. So as I indicated in the beginning, all of us is inviting 1 million participants across the United States to create this very large repository. And to date, there's over 178,000 individuals already enrolled. So they already completed their informed consent form, and they are in the process of linking their data, completing their questionnaires. But today, as a researcher, if you're wanting to propose and conduct a study using the All of Us research program data, there's already over 632,000 individuals. So it's a very, very large biobank. And what is very interesting is because of the process that they were working on recruitment, 84% of those individuals self-identify as members of communities that have been underrepresented in biomedical research. So it is really a gold mine for understanding variability in biology and understanding improving precision medicine that is for all, not only for specific populations and groups. All right, so let's talk about the data. So there's different data domains available in all of us research program. And then I include them in the slides. And of course, there's different varying degrees of participants that decided to share their data or to allow for genetic testing or to agree to share the electronic health record data. But both those are the components. We have survey responses, so all participants complete like very comprehensive lifestyle and overall health, social determinants of health. Participants also have information on physical measurements. So it's you can some participants can are invited for in-person visits to obtain height, weight, blood pressure, and some other measurements. We have over 400,000 individuals in the data set today that have genetic data available, both genotyping arrays, but also short-read whole genome sequencing. Almost 400,000 participants with electronic health record data. So I'm talking about historic records with diagnosis, procedures, vitals that goes all the way back from several years, even prior to their enrollment. So it's a very large longitudinal data set. We also have some individuals with structural variant data and also with long read whole genome sequencing. It's a smaller number, but it's increasing every data release. One data element that I'm very excited about and familiar with are Fitbit records. So there's about 60,000 individuals in all of us that have decided to share their own Fitbit data. And there's two populations of those individuals. One is what they're called BYOD, or bring your own device, which is, if you're a participant and you have a Fitbit and you decide to share your Fitbit data with all of us, you will obtain that data. But there's also some initial studies where they were giving Fitbit devices to all of us participants in certain areas to increase representability. And then also there is this Exploring the Mind task data, which is a smaller study that enroll among the participants already enrolled in all of us to perform certain cognitive tests in additional like mental health and emotional well-being surveys. So you have some additional information about that too, which can be very interesting as far as like task specific data, not only like survey. So some of the advantages and limitations of each one of those data elements, I think it's important to say. So we do for the surveys, you have a lot of questions that are based on validated instruments. Surveys will translate into Spanish, so it's not the goal again is to be representative. And in the middle, you can see the different items and questions that are available. There's really thousands of survey elements in this. Now, some of the limitations is that surveys and questions can be skipped, so participants are not required to complete all of them. And there is well documented disparities in who answers all the questions or not, which could be a problem as far as generalizability. As far the EHR data, electronic health record data, the data is available, can be available as far back as the 1980s, depending on the health system that the participants are in. You might actually have data since back then. The data is really well standardized. It's based on this what is called common data model that's called OMOP, so Observational Medical Outcomes Partnership. It is a way of organizing the data in a consistent way that makes it easier to aggregate data from different sites. So it's also makes it easier to pool data to perform analysis on that. So you have data on conditions, laboratory and measurements, medications, procedures. Is very and vitals like historical trends of BMI and weight and height and blood pressures and things like this. Up until the current release, clinical notes or imaging are not available, but I already seen a call that in the next release, they're going to start releasing some clinical notes, which I think it's going to be very exciting to further improve phenotyping of individuals in this cohort. Not all participants have EHR data available, and sometimes participants are part of different health systems, and only the data from the health system that has an agreement with all of us is going to be available. So that's a limitation with any sort of EHR data. And I think it's something that we just have to acknowledge, but understand that it could also incorporate some biases. As far as physical measurements, uh, those can be obtained on those in-person visits that I mentioned. It could be based on self-report for those participants that were involved remotely, or also complemented from the EHR. So you're talking about quantitative and curated methods, measurements of blood pressure, heart rate, height and weight, BMI from calculated from height and weight, hip circumference and waist circumference. Again, some of the limitations are based on the data that's just being shared through the EHR, not necessarily collected as part of a visit. So the self-reported measurements are only height and weight. And then, of course, collecting where you obtain this data has an impact on the validity of the data. If you're collecting this from a clinical setting versus self-report at home, there might be some variability that, there are ways to mitigate this with data analysis, but it can also be a limitation. So as far as the Fitbit data, again, this is historical data. So if somebody decided to share the Fitbit data, the data can go back all the way to 2009. There's those two types of Fitbit enrollment. It's either to bring your own device or participants that were provided devices. So we have both participants. So again, that can also reflect a little bit of what are the inferences that you can make with this data. But it does add another layer of multimodal data, which I don't think it exists in any other biobank. So there are some other biobanks that have, let's say, 7-day actigraphy data for measuring like physical activity and sleep, but that's only a one-time point 7 days. Like this, we're talking about participants that they may have over 5 years of continuous Fitbit use data. So that's very powerful to any sort of like longitudinal research. Here are some of the data points that you also obtain from the Fitbit data. And then, of course, there are some limitations. The data is proprietary from Fitbit as far as like how the algorithms work. So how they define steps in activity or how they define sleep is not necessarily publicly open and available. So there's limitations right there. Individuals have Fitbit data, even though in future releases they are working on integrating other wearables like Apple Watch data, which I think that's going to be also coming up on the next release. Individuals that don't wear Fitbit doesn't necessarily mean that they are not active or they don't have this data. It's just that it's not available to us, right? And then, of course, there are some other data elements that are, supposedly available within Fitbit, like nutrition or hydration, and those are not necessarily available. And then as far as the genomic data, it's one of the largest and most diverse whole genome sequences widely available for research. That's really a big plus. And then as of now, there's no gene expression or protein levels available. So we're really only looking at DNA and variation on the DNA. Although there was a recent call because participants that share some of the samples, there are some biosamples that are stored. And there was a recent call for proposals to propose studies to actually request those samples and do any sort of like biomarker assessment. So that's something that all of us is actively working on and trying to engage with researchers trying to do this sort of work as well. Um, and then, of course, some of the phenotypes might have like limited availability. So one big challenge on trying to define certain disease phenotypes that are only based on the EHR is that EHR diagnosis and billing, they are not necessarily done for phenotyping, they are done for billing purposes. So you have to come up with methods to create computable phenotypes that are validated to define a phenotype of interest. So then you can use genetic data to test and validate against it as well. So, and then not everybody decided to have their genomic data available. And of course, working with genomic data can incur larger computational costs. So the way that once participants kind of have this data available to all of us, either through responding service when they enroll or through connecting to those additional data sources that we talked about. You have, all of us and the institutions that are partnering with all of us work through the whole process of combining this data together, harmonizing it, cleaning it, and then making it available in what is called this curated data repository or CDR. So that's where the data lives. And if you want to know what data elements, specific questions that are available, you can search for them in the data dictionary that comes along with the curated data repository. But if you want to know more about what data is available there, you can always go to what is called the All of Us Research Hub, which is researchallofus.org. There, even without a registration or a login, you can explore the data. You can go on what is called the data browser and then just type in a specific data element or a question, and you can get counts of how many patients have a specific condition or have a specific data element. You can also explore other people's projects with this data. So if you have an idea, it's probably a good idea to type it in. Hey, is anybody working with this sort of disease or condition? And then you can even get their contact and collaborate if you want. You can also explore the publications that are available that have been published using this data and just like look for any updates and new data releases. And again, the research hub, you don't even have to have a registration. This is all publicly available data. Now, if you are interested in analyzing the data, then you have to register, sign up, and I'm going to show the steps in a little bit. But basically, you have access to what's called the Researcher Workbench, which will provide a platform and to organize your projects. So then that will let you build your data sets, build your cohorts, and then come up with analytical notebooks to analyze the data, and then you can report the results of this data in a publication later. So those are the steps that from the participant to the researcher and publication, right? So the participant decides to share their data, that all of us data and research center will process this data, make it available in an aggregated way through the data browser or on an individual level way to the researcher workbench that can only be accessed if you have the authorizations and the approvals. A researcher interested in working with this data have to register their project on all of us, and that becomes also public. So every new researcher wanting to analyze new data on all of us, they have to publicly describe their project that's become available in the data browser and so then others can see what you're doing. And then they can create projects in the workbench. And after they analyze, they can go ahead and publish and share the data. So there are three data access tiers that will really depend on the level of training that you obtain. So the public tier is available to everyone, and that's the data that's available in the data browser, which is just aggregated. So we want to know how many patients the all of us has, how many individuals with certain conditions, or how many what is the distribution of the blood pressure across all participants by sex or by age groups. That's directly available in the data browser. So there's no registration. You just go into the website and play around with the data that's already there. So that's great to determine feasibility and say like, hey, can I do this study? Let me take a look at some of the numbers and see if there's sufficient patients with this condition. Now, after you obtain approval, which is available to all the registered researchers, you have what is called the registered tier, which will give you individual level data of all like EHR, wearable survey physical measurements, and then also some level of de-identification, like you have like date shifts and some protection of the data that to minimize to maximize privacy protection. But if you perform additional training, you can also obtain data on the control tier level, which includes the genomic data, additional expanded demographic survey and EHR data, including some geographic level data, like a three-digit zip code level data as well. So those are the four steps to register as a researcher in all of us. So first, you have to confirm that your institution has an agreement with all of us. So KU and most of, I believe all or most of our partner institutions within Frontiers have an agreement with all of us. So step one probably is already completed for you. Then you create an account and verify your identity. You actually have to upload a picture of your driver's license and go through like login.gov to ensure that you have that verification. Then you have to complete a human subjects training that is specific to all of us. And that training will depend if you're doing the registered tier or the control tier. So you have to do additional training on the control tier data. Those have to be completed annually for you to continue having your access. And then finally, you have to sign this data user code of conduct, which just indicates several statements, and then you have to agree that you're not going to do anything wrong with this data. Okay, now I'm going to move on to just some of the studies that my group and others are doing here at KUMC using the All of Us data. And I think, we're spanning across this whole spectrum of like sleep medicine, sleep apnea, wearable technology and digital health, health behavior adherence, Alzheimer's disease and genomics, social determinants and disparities, and cancer in women's health. So I'm actually going to start with this first, I think that was probably the first paper using the All of Us data from Dr. Diane Mahoney from the School of Nursing here at UMC, where she was interested in social factors influencing the perceptions of health in cancer survivors. So she selected a cohort of about 1,200 participants who were ovarian cancer survivors, and then tried to determine if different aspects of like social satisfaction, general mental health, and general health were differently associated with specific social determinants of health. And what she found is that some of those unfavorable social factors were associated with worse overall perceived health. Some racial and ethnic minorities were less likely to be diagnosed at earlier stages of cancer. More advanced aggressive cancer presentations were observed in minority groups. And one of potential conclusion of the study is that addressing those social determinants could be essential to help improve cancer survivorship and reduce those disparities for ovarian cancer. So Dr. Mahoney's work was, very interesting and kind of like led some of the initial publications using the All of Us data. So the next publication that I'm going to talk a little bit about is from a study that was done by Sonia Mishra, which is the research analyst from medical informatics. And in this study, we're interested in understanding the differences in diagnosis of obstructive sleep apnea, which is a sleep disorder affecting billions in the world, and how those would differ for individuals that use the self-reported questionnaire about their self about whether they have been diagnosed with sleep apnea versus the rates based on the EHR. And then we found that some disparities. So we have more people in the all of us data set that self-reported they have been diagnosed with sleep apnea than the data that's being captured in the EHR. And that, could reflect just a little bit of that the data quality gap that we see. Not all participants may have EHR data, but also it aligns with the underdiagnosis rates of sleep apnea that we generally observe in the general population. One interesting finding of this study, though, is that Kansas, for some reason, was the state with the highest rate of obstructive sleep apnea. And I wonder if those were most being diagnosed within here the KU system. So we also try to understand some of those disparities on the difference between EHR and self-report diagnosis. And then Sonia did this analysis where she identified a large number of social determinants of health questions and try to group them using this clustering method that's called latent class analysis. And then she identified three specific domains about burden of healthcare access, perceived barriers to healthcare, and geographical and socioeconomic risk. And within those three domains, there were different classes of individuals based on how they responded to those questions. And one of the things that we found in the study is that participants with lower healthcare access, they were less likely to have an EHR documented sleep apnea diagnosis, which makes sense. Also, individuals at higher socioeconomic risk based on their geographic location were less likely to have received a diagnosis of sleep apnea, regardless if it is based on the self-report or on the EHR. And social risk burden may predict car seeking and formal diagnostic patterns. So therefore, we need to understand in trying to understand care for sleep apnea for all, from where those participants are coming from and whether there are specific gaps that are preventing us from obtaining a specific information about sleep disorders on that population. So now moving on to another kind of theme, and I want to also highlight the work that was done by Kashfi as well. So she was at the time that she did the study in my group, she was a high school student at Blue Valley School District, and we were interested in understanding healthy behaviors, right? So we identify a number of survey questions in the study that are relating to just overall health and lifestyle behaviors, like substance use or alcohol drinking, or whether they've been vaccinated for flu or COVID, if they go to their doctor frequently, if they go to their dentist frequently, and so on, right? So we also use that same clustering technique in those 21 health and lifestyle survey items. And then we try to identify whether there were patterns that could be observed. And we found those four patterns based on those questions. So about 8.4% of those individuals were classified as being unhealthy, where you have higher rates of some like substance use and lower rates of going to the doctors and seeing doctors. And you have those other groups like a generally healthy individual, a health conscious individual, which they have high healthcare utilization, but probably because they are aware of their health. And you also have some individuals that are skeptical about their medical treatments. And what we did is we compare those clusters of healthy behaviors with diagnosis of conditions across all of the spectrum of the conditions available in the EHR. And then we see some kind of results that are somewhat expected. We see that individuals in that unhealthy cluster, they were also much more likely to have like tobacco use disorder or major depression, anxiety disorder. And in some individuals on the health conscious group, they were more likely to also have some of those conditions, but also more chronic and metabolic conditions, including like sleep apnea or pain, and also some other conditions that you can see in that plot. So that's like an interesting observation of how lifestyle behaviors can relate to health at a large scale. So another study that was very interesting now incorporating some genetic data, which is it's being led by Quentin Mensah, which is an MD PhD candidate here at the School of Medicine. He was interested in the interaction between lifestyle factors and common genetic risk factors for Alzheimer's disease. So as part of his study, he identified a number of potentially modifiable risk factors that have been described as important risk factors through the Lancet 2024 Commission that are risk factors for dementia. And he identified individuals from all of us across the risk across all of those risk factors and identified what is called polygenic risk scores, which is a way of summing the common genetic risk from specific genetic variants that we know that have been reported to be associated with Alzheimer's disease in this case. And then what he found is that if you classify individuals based on their genetic risk, which are those colors, and based on their risk factor based on the modifiable risk factor scores as favorable, intermediate, and favorable, what we see is that the modifiable risk factors have a much greater impact on the risk for incident dementia compared to the genetics, but there is a small moderating effect. And then again, I don't have this on this slide, but there's a very important common genetic risk factor for Alzheimer's disease, which is the genotype, for a specific genotype on the ApoE gene. They actually contribute independently in a much larger effect than the polygenic risk for effect, which is a little bit more modest. So one of the conclusions of this study, which is currently under review, is that the Alzheimer's disease risk is driven more by modifiable risk factors and ApoE genotype than the polygenic risk score that was obtained in this study. So another study that has been published using wearable data, and that was just a shout out from past Frontier CL1 scholar, pre-doctoral scholar Colbert. So he analyzed the wearable data, the Fitbit data from all of us, and trying to determine whether surgery for chronic rhinosinusitis could actually impact participants' steps before and after. So would participants become more active after they treat their chronic rhinosinusitis with a surgical intervention? And what they found, even though they only had a small number of individuals in the data set that went through this procedure, because you have so many data points as far as how the data has been collected longitudinally for Fitbit, that daily steps were actually higher and more time spent on vigorous activity after surgery in the surgical cohort when compared to the control cohort. So this, they claim that that's the first wearable derived objective evidence of improved real world physical activity following sinus surgery. So very interesting. And again, a shout out to Cole. And then I think the last study that I have to share here, which is something that we recently presented at the Sleep meeting a couple weeks ago, is going even further on this understanding how we can use wearable data. And since we're looking at longitudinal data for several years, we can probably understand some of the patterns that we're generally people are curious about. So do we see an association between sleep and whether weekends and work days, or is there a seasonal trend across the year as far as how much you sleep, or relationships with age, right? So we looked at this data, which pretty much compiled every single night of available data across all the individuals in all of us. So we're talking about here about over 22 million nights, over 42,000 participants. And then we do see, expected differences where people tend to sleep slightly longer in free days compared to work days. We do see a seasonal trend on sleep duration. So people tend to sleep less during the summer here in the United States and more during the winter. And I think that tracks along with the availability of daylight during the day, and particularly daylight towards the end of the day, we tend to see a drop in sleep duration with age. And again, that's highly documented. And also the midpoint, which is that midpoint between the average sleep onset, so the time you go to bed, and the average sleep offset, the time that you get off bed. The midpoint is just like a measure of how early or late your sleep is. It's also decreases a lot with age, and again, something that is expected, but it was nice to demonstrate not only based on a single week of an actigraphy use, but across the board. But what I think I'm more excited about this study is that in the United States, there are some participants, there are some states that do not adopt the daylight savings time. So Arizona and Hawaii are states where there's no daylight savings time transitions. They are all on permanent standard time all year round, while the other states have the variability. So using the Fitbit data, we were able to demonstrate the impact on the sleep midpoint of those states where you do see the daylight savings time transition. So as expected, you see almost like a 1 hour shift on the sleep midpoint because of the daylight savings time transitions. And we don't see a major effect on sleep duration. So we believe that there could be some other mechanisms that are impacting that are based on, chronotype and just like alignment that we can observe from this. So, and then we are also analyzing a little bit more details around the spring forward and the fallback periods, which are when that shift occurs more abruptly, since it has been documentation in the past that during those transition times, there are also greater risk for some like incident cardiovascular events. So some more to come on this data that we're actively analyzing as well. Okay, so now to finalize my presentation, I just want to kind of share a little bit more on opportunities to get involved. So if some of those studies and the process intrigued you and caught your attention, are the only one. There's over 21,000 registered researchers across over 1,300 institutions, and there's over 24,000 projects available in the workbench, and now more than 1,200 publications available. One research dissemination effort that I would like to highlight here from KUMC was this paper that was published by Dr. Daniel Parente at Family Medicine, where he pretty much wrote a description of the All of Us data set focused on primary care research. So if you're interested, highly recommended manuscript, especially to get kind of like started. And the other dissemination efforts that we're having here is training. So if you're interested in learning how to use this data, we are then offering this training program, which is called TODAS, Training Observational Data Analysis Using All of Us, every spring. So we just concluded our first class, our first session, this spring, and last week is the last lecture. But we had a great turnout, and we, went through the whole process from understanding the data, designing your study, and then implementing the study in the researcher workbench, all the way to obtaining your results. So that's that. I think that was a very successful experience, and I had a lot of fun teaching this, and with the help of the collaborators that I have listed here on this slide. Now, what about you as a participant? And I think, as I said in the beginning, I'm a law of us participant, and I think that really helps us to understand the impact of the project, right? So all of us only really works because it's meant for all of us, right? So if we can do this, if we can access this data as researchers, imagine if you can actually be part of it as a participant that is also contributing the data, that's incorporating the own realities and variabilities of your own, your own individual into this data set. So it is a chance for you to be engaged with the community of individuals that are, visionaries in understanding research, and also your opportunity to learn more about other research opportunities. So if you want to, either spread the word about a researcher, how to use this data as a researcher, or to join as a participant, you have some QR codes here on the screen. But we also have some of the team here from the All of Us research, All of Us Heartland Consortium that can help you guide the right resources, or you can also directly contact me. With that, I would like to thank you all for this and thank you all my collaborators and everybody that's been extremely motivated to be engaged with this data set from the participant perspective, from the researcher perspective, from a training perspective. And I am very excited to, just be part of this project and help you achieve your goals either as a participant or as a researcher. So thank you. I'm happy to address any questions that the group may have. And we have Nikki Malomor here that can also help some of the questions with joining as a participant as well. Thank you very much.
Thank you. That was a really excellent overview. I've heard a lot about All of Us, but now I feel like I really kind of understand what all is incorporated into it and kind of all the ways that it could be useful. So those examples you provided were really helpful. So I will open it up to questions. If anyone has questions, you can actually unmute and ask your question, or you can put it into the chat. I don't see any right off. I did have one question. Well, okay, here's one. I will hold mine for now. So, Dr. Shabana John asked, could you tell me when the next training session is from your department?
Yeah, we'll probably launch that next spring now. So I'm anticipating it's going to be somewhere around between March and April next year.
Okay. And do you know, will that be something that that will be available to those outside of KU Medical
Yes,
Center?
yes.
So Frontiers?
This is open. Like in this first session, we actually had folks from all over the US, from the West Coast to the East Coast. And we do, I mean, there's lots of questions about like just access and recordings, and we do have the recordings available, but it is a little tricky because a lot of the material that we cover goes over individual level data, and you actually have to have all the permissions to access the data in order to be able to see that data. So the access to the videos for those that are not enrolled in the program is a little bit more limited. But if if for some reason, you feel like a course like this is not going to be something that you have the ability to join, it's about like 2 hours a week for 12 weeks. So it's not a lot, but it's not a little. So it's something that if you have access to your to the workbench, your control tier user, you can reach out and then we can just I can share some of those recordings of the validation that you can actually see the data pretty much.
Okay, thank you. And hopefully, once those dates are set, if you could share it with Frontiers and we can share it out so that the Frontiers partners can access it.
Yes, absolutely.
Thank you. And then I think we had another question about, can you talk a little bit about the costs associated with data use? So is there a
Yes.
cost?
Yeah, so there's no cost to access the data. What really costs money is analyzing the data because you have to launch some like cloud based systems, and that cloud
Mm.
computing costs money. Now that said, every new user to all of us, they receive $300 in initial credit, and that initial credit is more than sufficient to run many studies. So my credits recently expired and I didn't spend $200 of them. And I have so many ongoing studies with using all of us. Granted that, none of the studies that we are working on within my account are genomic studies. So those will cost a little bit more, like they will spend a little bit more credits. But even then, it is not, $300 gives you a lot of room to play with. Now, one recent thing that all of us did is that those $300 initial credits, they expire. So they didn't used to expire before. Now, after 1 year, they expire. So, you might as well spend them as much as you can. Now, there are some processes. I think KU has already signed some contracts with a contractor that helps us with Google Cloud platform storage. So after your credits expire, if you have like a source of funding within your own department, it's actually straightforward to connect that funding line with the all of us. So then those get billed to you. So and that's really going to depend on how much computing you're using. So based on the top of my head, I think it costs about 10 to 20 cents per hour of actually the computer that's running. So of course, it's only running when you're logged into your session or you let it run. So it's not really a lot. And then there's an additional $5 per month for storage costs. Although with the new researcher workbench, they're recently transitioning from like a 2.0 version that's going to be slightly different. So I don't have the new costs on top of my head, but you can probably find that online.
Okay. And then there's another question. Do we have to follow any IRB protocol to access the data? And then for NGS data, which system needs to be followed to access and analyze the data? And then can I access the data as a postdoc researcher, or does it have to be through a PI?
Yeah, so those are great questions. So the data use agreement should cover the IRB, although it is always recommended that you have your own internal IRB. That's going to be a secondary data use. So those are generally like, some institutions see that as a waiver, but it's always recommended that you have like your IRB for ongoing studies with any sort of like secondary data analysis. For the next generation sequencing data set, so the available data, you can obtain like VCF files. So if you're familiar with some of the nomenclature that's behind, so foreign call format files, those are directly available. They also have a specific format that's called it's from like a package that's called like Hail, which is it's a new, more efficient method for analyzing genomic data, and they provide lots of tutorials. So the data is available in that format, but there's a lot of like, example codes and tutorials on how to use that data. And yes, you can access the data as a postdoc. It doesn't have to be a PI. That really the criteria is that you have to be affiliated with an institution that has an agreement with all of us. So if you have a like an email account from an institution that has an agreement with all of us, you should have access to the data, assuming that you complete all the steps of registering, completing training, and so on.
You. And then we have another question here. Is there any timeline that they follow to release more data? And then I also had a question kind of along those lines as far as is any of the data prospective other than the EHR and Fitbit data?
You mean prospective or
Yeah, or will
retrospective?
they be doing additional surveys along
Oh,
the way or
yeah, so they do have a roadmap. I don't believe that it is like every couple of years they release new data like that. But after you register, you receive like newsletters and emails that always update you about the next step. So that's when I mentioned something about, oh yeah, I think the Apple Watch data and clinical notes data will become available. It's because they usually release that. So I don't know, and I'm not sure, Nikki, if you have any information about their next data release, but I believe once you register to the system, then you get access to all of that up-to-date information. And another interesting thing too is that they offer weekly office hours. So if you have any questions about anything, all of us related, you can join those office hours and then you can ask those questions directly as well.
so do you get plugged into those office hours once you sign up for the researcher
Yeah.
workbench?
Yeah.
Okay. All right. Lots of great questions. We have 1 minute left if there's maybe one more, but if not, I think that was really excellent. Thank you for the overview, and we look forward to learning a little bit more when the new training sessions come out.
Yeah, great. I appreciate it. And if you have any questions, feel free to reach out and I'm happy to chat with you about it. Thank you.
Okay. All right. Thanks, everybody.
All
Have
right.
a
Bye.
great rest of your day.